Imagine, if you will, being able to literally sit down and just have a conversation with a cancer tumor.
Right, yeah.
Like, what if you could just ask it, point blank, exactly how it's managing to evade the immune system, and it actually answered you back in plain, understandable English?
I mean, it sounds like pure science fiction, honestly, like a biological cheat code.
Yeah.
But the reality of what researchers are building right now is rapidly turning that thought experiment into a tangible tool.
Yeah, and that is exactly why we are taking a deep dive today into the blueprint for what the scientific community is calling pharmaceutical superintelligence.
That's a heavy term, yeah.
It really is. We're looking at an incredible stack of sources here. We've got research papers on AI architectures, spatial multi-omics, and multimodal learning.
Mm.
And the mission here is to figure out how scientists are pairing raw, incredibly complex tissue data with an AI interpreter called CellWhisperer. And the end result is actionable natural language biomedical reasoning.
Right, because the overarching goal, the entire reason this architecture is even being built, is to drastically accelerate drug discovery.
Right.
We're moving toward a reality where autonomous AI agents can seamlessly decode the physical architecture of a tumor and identify therapeutic targets entirely on their own,
Right.
completely bypassing the human bottlenecks that, currently slow down medical research.
And whether you are a biotech professional who, lives in these data sets every day, or you're simply someone fascinated by how the future of medicine is unfolding, this deep dive is going to show you a massive shift.
Absolutely.
We are moving past AI that just crunches massive spreadsheets of numbers. We are looking at AI that actually understands the physical reality of biology and can talk to us about it.
Exactly. And to reach that level of superintelligence, the AI first needs to understand the tumor not just as a random list of mutated genes, but at a physical place.
It's like a real 3D space.
Right? A living, highly structured ecosystem.
Mm-hmm.
And historically, that has been incredibly difficult. The traditional method for understanding a tumor's genetics involves something called single-cell RNA sequencing.
Okay, right.
But the major flaw in that process is that it requires dissociating the cells. You have to literally use enzymes to break the tissue apart so you can read the genetic material of the individual cells.
So if single-cell sequencing requires breaking the tissue apart, I'm trying to picture this. It's almost like taking a whole city and putting it in a giant blender.
That's exactly it.
You analyze the resulting smoothie to find out what the average person in that city is like. You might figure out the city has a lot of bakers and a lot of teachers, but you have zero idea who lives next to whom.
That captures the limitation perfectly. You completely destroy the physical context. You lose the map entirely.
Wow.
But a tumor is an architecture. The cells rely on their physical proximity to each other to survive and grow. This is where our sources introduce spatial multi-omics, which completely changes the paradigm.
Spatial multi-omics.
Right. Instead of blending the tissue, spatial multi-omics profiles the transcriptomics, the proteomics, and the metabolomics directly within the intact tissue.
Hold on. For those of us who don't have a PhD in molecular biology, let's just ground those terms real quick. We are basically talking about the cell's instruction manuals, their physical building blocks, and their energy output, right?
That is a very clean way to look at it, yeah. Transcriptomics shows us the instruction manual, which genes are turned on or off.
Okay.
Proteomics shows us the building blocks, like the actual proteins being produced based on those instructions. And metabolomics shows us the energy and chemical processes keeping the cell alive.
Got it.
So spatial multi-omics maps all three of those layers at once, without moving the cells from their original physical location.
So instead of the blender, we now have a high-resolution satellite map. We can see the bakers, we can see the teachers, and we can see exactly what neighborhood gossip they are spreading.
Exactly. And in oncology, that neighborhood is called the tumor microenvironment, or TME.
The TME, right?
Right. Spatial omics tracks how cancer cells, immune cells, and stromal cells, which are essentially the structural scaffolding cells that hold the tissue together, how they all organize themselves. The research refers to these organizations as functional niches. They're these specialized spatial communities.
Why does that physical location matter so much to the AI, though? I mean, does a cancer cell actually change its behavior just based on its zip code?
Oh, it fundamentally changes its behavior. The sources detail specific spatial phenomena that we can only see if we keep the map intact.
Like what?
For instance, they identify leading-edge phenotypes. These are highly specific organizations of cells at the very boundary of a tumor. Because they are on the edge, they adopt distinct behaviors to drive metastasis.
Oh, I see.
Yeah, they are the vanguard actively pushing to spread the cancer to new tissues.
So the vanguard of the tumor acts completely differently than the cells hiding safely in the center.
They do, entirely. Another crucial example the papers highlight are hypoxic and perivascular niches.
Hypoxic meaning low oxygen.
Exactly. These are areas deep inside the tumor with very low oxygen, or areas wrapped tightly around blood vessels. The spatial mapping reveals that tumors specifically use these exact physical geographies to foster immune evasion.
Wow.
The cancer cells essentially use the terrain to hide from the body's immune system.
That's terrifying but fascinating. We are mapping out the enemy's fortresses, their vanguard, and their supply lines. But here's where I get stuck.
Okay, what's that?
We now have this incredibly complex, high-resolution satellite map of the tumor. But the data on that map isn't a picture, right? It is written in raw, high-dimensional RNA sequences and protein expressions.
Right, it's massive.
It's a massive, incomprehensible ocean of molecular coordinates. How does an AI agent actually read that and make sense of it?
Well, it requires a highly specialized translator. The spatial map is useless without an interpreter, and that is exactly the role of CellWhisperer.
CellWhisperer.
Right, it acts as the bridge between that raw spatial data and actual human language.
Now, the paper explicitly states that CellWhisperer allows the AI suite to chat with single-cell data using natural language. And I need to push back here because, we've all seen language models confidently hallucinate facts.
Oh, absolutely.
So is this AI literally translating raw RNA sequences into English? How do we know it isn't just making up biological stories that sound plausible?
It's a fair question, but the architecture of CellWhisperer is specifically designed to prevent that. It is a multimodal AI model, meaning it learns from multiple types of data simultaneously.
Okay.
It uses a technique called contrastive learning to integrate RNA sequencing profiles from a model called Genoformer and biomedical text from a model called BioBERT.
Genoformer and BioBERT.
Right. And it forces both of those data types into a single joint embedding space.
A joint embedding space. How does that actually work mechanically? Like, how do you force a genetic sequence and an English paragraph together?
Imagine a massive multidimensional room. When the model is training, it takes a raw transcriptome, the RNA profile of a specific cell, and the verified, peer-reviewed textual description of what that cell is doing.
Okay.
It places them both in this room. Contrastive learning works like a magnet. If the RNA profile and the text describe the same biological reality, the model pulls them closer together in that mathematical space.
Oh, I see.
If they describe different things, it pushes them apart.
So it's basically building a bilingual dictionary through physical proximity. RNA is on one side, English is on the other, and the model binds them together.
Exactly. And to give you an idea of the scale required to build that dictionary, the sources note the model was trained on over 1 million transcriptomes, paired with AI-curated description.
1 million?
Yeah, 1 million pairings from massive databases like the Gene Expression Omnibus and the Cellus Gene Census.
That is a staggering amount of vocabulary practice. But does it actually hold up under scrutiny? Like, does it work?
The metrics are incredibly strong. The model achieved a mean AUROC score of 0.927 for retrieving matching transcriptomes from text.
Okay, let's break down AUROC for a second. What does a 0.927 actually mean in practical terms? Because that sounds like math jargon.
Fair enough. AUROC stands for Area Under the Receiver Operating Characteristic Curve. In plain terms, if you give CellWhisperer a random RNA sequence and a mix of textual descriptions, 92.7% of the time, it will correctly rank the true matching description higher than a random one.
Wow, so over 90%.
Yeah, it is incredibly accurate at looking at an RNA sequence and retrieving the exact correct English translation.
So it's not hallucinating because the English text is mathematically anchored like a tether to the actual molecular data.
Exactly. And the applications of this are staggering. Because of this architecture, autonomous agents in the broader AI suite can actively interrogate these spatially identified cell clusters.
Interrogate them, like question them.
Yes. An agent can basically look at a cluster of cells hiding in one of those low-oxygen niches on our spatial map and query it in natural language. It can ask which genes are highly expressed in these cells, or describe this subpopulation of cells in detail.
We went from blending tissues in a tube to pointing at a specific neighborhood on a cellular map and asking an AI, hey, what are these guys up to? And it replies, Oh, they're currently upregulating stress response genes to build an immune-evading fortress.
That's literally what's happening. And it
Right.
formulates that answer based on the raw, high-dimensional data it is reading in real time, not just by reciting a pre-written textbook.
That is wild. So if this AI is building a bilingual dictionary for biology, what happens when it encounters a word or, a mutated cell that it's never seen before
Right.
because a tumor is constantly mutating? How does the AI handle a cellular behavior that just wasn't in its training data?
That brings us to one of the most powerful capabilities detailed in the sources, which is zero-shot phenotyping.
Zero shot.
Yeah. Because CellWhisperer understands the underlying fundamental rules connecting RNA to biological concepts, it can predict cell types, tissues, and disease states from spatial transcriptomes without needing to be explicitly trained on that exact variation.
Zero shot, meaning it can just figure it out on the first try. Because historically, if you wanted an AI to identify a rare immune cell, didn't you have to manually feed it an extensive, human-curated database of markers for that exact cell?
You did.
Yeah.
And creating those databases takes years of manual laboratory work. CellWhisperer completely eliminates that
It
requirement.
just bypasses it.
Right. It can accurately phenotype cells it has never explicitly seen labeled before by inferring their function from that joint embedding space.
If you're listening to this and you've ever had a loved one wait wince for a tumor biopsy result just to figure out the right chemotherapy protocol, this is where this technology gets deeply personal.
It really does.
We're talking about reducing weeks or months of manual human lab work down to instantaneous AI analysis.
It compresses the timeline dramatically. And it goes further than that with in situ target validation.
What does that mean in practice?
Well, let's say the spatial omics map reveals a genetic alteration happening live in the tissue. For example, during tracking minimal residual disease, where we are watching how a tumor evolved over time to resist a drug.
So we're literally watching a mutation happen geographically inside the tumor.
Exactly. When that happens, CellWhisperer analyzes the transcriptomes of those specific mutating cells. It automatically identifies novel marker genes associated with that new mutation and evaluates their biological roles.
Wow.
It can instantly output this mutation is triggering a massive stress response, or this alteration is driving an extracellular matrix remodeling program.
So it is essentially performing the scientific method live inside the tissue. It observes the map, translates the data, forms a hypothesis, and hunts for the therapeutic target.
Yes,
But the sources point out that CellWhisperer is just the interpreter. To actually achieve pharmaceutical superintelligence, this translated data has to feed into a larger system, right?
exactly. CellWhisperer is the crucial foundation. But the apex of this technology is feeding that highly refined, validated intelligence into a decentralized multi-agent system.
Okay, a multi-agent system.
Right. Our sources outline a theoretical framework called the Darwin-Gödel machine, or the DGM. Achieving true superintelligence requires distinct, specialized AI agents to process this complex biology, form abstract concepts, and communicate those concepts to each other seamlessly.
I want to understand this conceptual communication because we aren't just talking about one giant monolithic brain doing all the work here. It's a network of specialized agents. How do they actually talk to each other about a complex biological perturbation without losing the nuance?
They do it by compressing high-dimensional data into what the research calls concept vectors.
Concept vectors.
Yeah, when specialized agents analyze a newly mapped spatial ecosystem, they use CellWhisperer's joint embedding space to form these abstract vectors.
Let's try to visualize that. Is it almost like taking a 1,000-page medical textbook on a specific cellular mutation and mathematically compressing all of that knowledge into a 3-word summary that another AI can instantly read and completely understand?
That's actually a brilliant way to conceptualize it. The sending agent compresses the biological reality into a highly dense mathematical vector, and the receiving agent unpacks it using their shared symbolic interface.
So they can transmit entire biological concepts instantaneously.
Exactly.
Wait, so if these specialized agents are forming their own biological concepts, compressing them, and transmitting them across the network, they're basically teaching each other. They are skipping the human learning bottleneck altogether.
They are continuously educating the collective network. However, to ensure they aren't just trading highly convincing hallucinations back and forth, these transcriptomic insights are fed into a neurosymbolic architecture called GraphMERT.
GraphMERT. Okay, neurosymbolic architecture. We need to define that. Is that like combining the pattern matching of a neural network with the strict rules of symbolic logic?
Precisely. A neural network is great at finding hidden patterns in massive data sets, but it struggles to explain its reasoning.
Right.
Symbolic logic operates on strict, verifiable rules and facts. GraphMERT combines them. It takes the newly discovered spatial marker genes and anchors them into verifiable causal knowledge graphs. It acts as the strict scientific fact-checker for the entire multi-agent system.
But how strict is this fact checker, really? Because the sources mention GraphMERT achieves nearly 70% factuality. I have to admit, if we are talking about superintelligence designing drugs that will go into human bodies, 70% sounds like a failing grade.
It sounds low at first.
Why is that considered a breakthrough, and what happens to the other 30%?
It's a vital question. But you have to compare it to the baseline. In traditional large language models asked to perform complex, multi-step biological reasoning, explicit causal accuracy, where the model can mathematically prove every step of its logic, is incredibly low.
Ah, okay.
GraphMERT achieves nearly 70% factuality with explicit sentence-level provenance.
Sentence-level provenance, meaning it can point to the exact line in a peer-reviewed paper or a verified dataset to prove its causal chain.
Exactly. For 70% of its complex reasoning, it provides the exact, verifiable proof. The other 30% isn't necessarily a hallucination. It may just be a probabilistic inference where the exact literature citation is missing.
Oh, I see.
But the crucial part is that the system knows the difference. It ensures the autonomous agents rely on verified axioms for drug design, not probabilistic guesses. This level of verifiable mechanism of action is completely non-negotiable for eventual FDA approval.
So the system flags the uncertainties and relies heavily on the proven axioms. We have CellWhisperer translating the spatial data, the agents forming concept vectors to teach each other, and GraphMERT rigorously checking the math. How does this cycle actually result in new therapeutics, though?
Through continuous recursive refinement.
Recursive refinement.
Right. With CellWhisperer constantly translating new spatial measurements into queryable text, the Darwin-Gödel machine is fed a never-ending stream of domain-specific intelligence. And the system uses this constant influx to engineer what the sources call synthetic polymath primes.
Synthetic polymath primes, that sounds incredibly imposing. What does that actually look like in the system?
Think of them as strategically engineered, highly efficient meta-training models. The system basically fuses its models of chemistry, physics, and biology together.
Okay.
When it discovers a new spatial marker or a new immune-evading tactic, it engineers a synthetic polymath prime to update the entire system's understanding of disease mechanisms. It is an open-ended, self-improving cycle where the AI transcends human-designed architectures to autonomously design precision therapeutics.
It's basically writing its own multidisciplinary textbook, constantly updating it, and using that knowledge to formulate cures for diseases that currently baffle us.
It is the absolute definition of a paradigm shift.
Let's pull all of this together because the journey we've gone on today is staggering. We started with the physical intact architecture of a tumor using spatial multi-omics to map the exact neighborhoods, the functional niches, and the vanguards of the tumor microenvironment.
Right, moving from the blender to the high-res satellite map.
Then we explored how CellWhisperer acts as the interpreter. It uses a joint embedding space to translate that raw, high-dimensional RNA data into natural language, proving it isn't hallucinating by anchoring it to a million sample foundation.
Which unlocks the ability to zero-shot phenotypes and hunt for therapeutic targets in real time, observing mutations as they happen live inside the tissue.
Exactly. And finally, we looked at how all of that validated intelligence feeds into autonomous systems like GraphMERT and the Darwin-Gödel machine. Networks of specialized agents compressing data into concept vectors, teaching each other, fact-checking each other with explicit provenance, and engineering synthetic polymath primes to recursively evolve their own biological understanding.
The core takeaway for anyone listening to this deep dive is that the future of medicine is no longer about just collecting more data. We already possess mountains of raw data.
Right, we're drowning in it.
The future is entirely about systems that can autonomously reason through that data. It's about an AI collective that can look at a tumor's physical architecture, fluently understand the hidden language of its cells, and proactively design precision therapeutics faster than humanly possible.
We started this deep dive by imagining what it would be like to sit down and have a conversation with a cancer tumor in plain English. And the reality is we are actively building the exact technological translator needed to do exactly that.
It's happening right now.
But knowing how this multi-agent system operates, it leaves me with one final lingering question for you to mull over.
Hmm.
If systems like the Darwin-Gödel machine are autonomously engineering their own synthetic polymath primes, if they are discovering novel spatial markers and compressing them into concept vectors that bypass human learning entirely, at what point does the AI's understanding of human biology become so advanced, so fundamentally complex, that we actually have to ask the AI to dumb down its cures just so human doctors can understand how they work?
Wow, it's a profound thought. The exact moment when the translator becomes vastly more knowledgeable than the listener.
Definitely something to keep in mind the next time you look at a medical scan. The tumor is talking. We just finally learned how to listen.